A cellular filter decides what you remember and what you forget. Think of your cell's endosomes as tiny sorting centers, constantly deciding which signals get recycled back to life and which ones are sent for destruction. This isn't just molecular housekeeping, it's how your body archives existence itself. The architecture dictates awareness. Whether a receptor signal stays strong or fades away depends entirely on the kinetics of its journey through this internal pathway, a complex ballet managed by proteins like RAB-GTpases. It's more profound than simple downregulation. It is active, real-time memory editing happening beneath your skin. Your capacity for adaptability isn't fixed, but constantly being sorted and rewired at a microscopic level. We often think of the brain as receiving signals, but what if perception is actually a process of sophisticated cellular sorting? Think of your cells like incredibly complex city transit hubs. External information, every stimulus you encounter, arrives on these receptors and must be processed immediately. Some signals are critical. They need recycling back to reinforce existing paths. Others are noise that needs efficient disposal or downregulation over time. This intricate traffic flow is managed by specific molecular gatekeepers known as RAB-GTpases. These tiny regulators dictate whether a signal fades away in the lysosome or becomes permanent through dynamic cellular reshaping, defining how your system remembers and adapts moment by moment. Think about how your brain manages incoming data. It's not just receiving, it's aggressively sorting and discarding information based on necessity. Our research shows that this happens at an even smaller scale, inside every single cell. Cellular trafficking isn't random recycling. It is architecture dictating consciousness. These molecular pathways function like a hyper-efficient filter for reality itself, deciding what signals stick around to shape who we are versus what gets swept away and forgotten forever. Which means something nobody expected for how quickly adaptation can happen when you change the rules of cellular filtering. Think of your cells not as passive receivers, but as dynamic librarians deciding what information to keep or discard. Every time a receptor captures an external signal, a bit of light, a stressor, maybe even a chemical cue, the cell must decide its fate: recycle that knowledge for continuous function or send it to the lysosome for permanent forgetting. This process isn't random sorting, it's active architectural filtering governed by molecular traffic cops like RAB-GTpases, determining if an experience is temporary noise or stable memory. But some researchers argue this architecture is too rigid, suggesting a static blueprint limits true adaptability. Who has truly found evidence of complete cellular fluidity? You need to see how these tiny recycling kinetics govern your biggest shifts in state. The cell receives a message, like picking up a crucial piece of intelligence from the environment. That signal is internalized by tiny vesicles, the endosomes. But this isn't just storage. It's quality control on a microscopic level. These structures actively decide what matters and what needs to be forgotten. The way receptors are sorted here determines whether an outside influence sparks lasting change or simply fades away, recycled back into the membrane. It is a self-organizing field deciding reality itself. What happens next, the precise molecular choreography of that decision, is where perception shifts entirely. We have always viewed these internal cellular sorting points as simple mechanisms for disposal or repair, like a municipal recycling plant taking in materials and deciding whether to toss them or reuse them. But the architecture is far more complex than mere logistics. Think of it less like a bin and more like a sophisticated lens that filters all incoming information before it ever reaches your awareness. The way these internal routes are built dictates not just what a cell keeps, but how deeply it remembers it. This dynamic, self-organizing field, the endosome itself, is the blueprint for perception. And this is where everything shifts. What this all means is that your ability to adapt isn't just about willpower. It's rooted in a dynamic sorting system inside every single cell. Imagine the endosome as an internal cellular triage desk, constantly deciding what signal stays and what gets filed away forever or sent for recycling. The way we perceive stress or focus reflects this molecular filtering process. If you are overwhelmed, your internal architecture is telling you that something needs to be downregulated, not just fought. Pay attention to where signals flow in your body because that tells you how your system organizes itself. Understanding the principle of cellular plasticity, the ability to recycle and adapt, gives you a map for reclaiming mental resilience right now. The full research archive is at jmthomasofficial.com. The cell is not a machine, it's a filter. Every incoming signal, Every bit of information from your environment is run through internal sorting protocols, deciding whether to recycle that lesson or discard it entirely. Think of this like an architectural blueprint for attention. When we understand these molecular traffic flows, we gain insight into how our own focus is being allocated and what signals are truly making the cut to shape who we become next. True adaptability comes from consciously governing your internal sorting mechanisms. The full research archive is at jmthomasofficial.com.